Good afternoon, everybody. I really wish that I could be physically here with all of you, but let's hope this time next year we'll be all be in a very different scenario. And first of all, I want to thank all of the organizers for really just putting the show on the road. This has been exciting, a very different approach to how we can discuss comparative genomics, how we can really look to nature to find solutions to some of our problems. And what I want to do today is I want to introduce you to what I think, and many of us who are part of this group, is the most fascinating clade of all mammals. And these, of course, are our bats. And this lovely image looking at you, this beautiful creature, if you notice its sharp teeth and big long wings, this actually is a vampire bat. So, yes, we're coming near to Halloween. We're in October. And of course, here's a real vampire bat. So what I want to do today is I want to talk to you about BAT1K, what BAT1K is, what we hope to do, what we achieved so far, and where we're going to take to the next level. We have some wonderful speakers lined up to talk a little bit about what we can do with BAT genomes. But really, if you think about and look at this Earth Biogenome Project, we want to sequence every eukaryote species on this planet. And we have to decide, well, which lineage, which group do we want to study? And I'm going to say that we should all be looking at bats. Well, let's think about why. They're the only mammal that have achieved true self-powered flight. So the only mammals that fly. They have evolved really unique adaptations that allow them to have this extended lifespan. It's pretty hard to find a bat that has cancer. They have ways to resist it. They have extraordinary innate immunity and extraordinary immune responses. And we know right now that's very important. And I believe that studying bats can allow us to find novel treatments to dealing with viruses and diseases, potentially such as COVID-19. They're really important for our ecosystem services. We need them to be able to act as keystone predators and feed on insects and different types of crop and pests. But also they're very important for pollinating. So bats as an order are very important. 1 in 5 of every living mammal today is a bat. So they're extraordinary, they're unique, they have amazing diversity. And to try and understand this diversity, again, here is an example of the world's smallest mammal. This is Crassionyctus thonglongii. These are the fingers of my very first PhD student, Sebastian Puschmeier. So this wonderful, extraordinary radiation of mammals, we want to try and understand it. And to do this, I was involved with Genome 10K from the very beginning, with the Vertebrate Genome Project, with now the Earth Biogenome Project, with the Sanger Tree of Life Project, but we're leading the bat charge. And myself and a group of very interesting people, and this is the other co-founding director of BAT1K is Sonia Vernies. We have our executive members. We have David Ray from Texas Tech in the United States. We have Michael Hiller from the Max Planck and now in Segenbergen. We have Jean Myers from the Max Planck in Germany, Liliane Davalos for Stony Brook in the USA. So we pretty much came together initially and thought, hey, wouldn't it be a cool idea to sequence the genome of every living bat species? And together we came and we cobbled together these ideas, and we did this essentially on a shoestring with very little funding, but thankfully Jean was able to secure money from the Max Planck. And we got together with this steering committee of wonderful researchers in lots of different fields of bat biology. And we came up with our motherhood concept. What do we want to do? We wanted to sequence the genome of every single living bat species. Approximately 1,400 species are recognized today. And we wanted to sequence their genome to chromosome level error-free assemblies so that we can uncover the genomic basis of their unique traits and their extraordinary adaptations. And ultimately, we want to drive a global awareness of their conservation and their different issues that trying to study and work with bats can pertain to the world. So what we've done so far is, I don't know if you saw the cover of Nature in July, we were able to publish our pilot study where we sequenced the genome of 6 different bat species representative of the unique traits that bats have from all over the tree. And together we came up with the ideas and the methodology of how do you sequence these genomes to chromosome level assembly. working again with VGP and with other members of this community. We came up with the science behind how you analyze these genomes. And we use these genomes to uncover their unique adaptations, their extraordinary innate immune system, their adaptations of longevity, among others. And what we're looking today is we really want people to join us. So please sign up to BAT1K, become a member. And we had a wonderful Batellite, aka a BAT satellite meeting last night. And we spoke to the community about what do people want? What do we need to join? What do people want to see if they were to join BAT1K? Well, really, we're starting a new Slack channel. We're going to have more of these interactive forums because it seemed to work really well last night. So if you're interested in any of these unique traits that bats have, please join us. And what we're going to do today and what we're going to hear, and I'm very excited about this, are these 4 very diverse talks that really represent the diverse area of research you can be involved in studying bats. And what we're going to do is we're going to play these pre-recorded talks back to back. We're going to have a question and answer discussion section at the end. And so what I'd like you guys to do is, as the talks are happening, write at Zaja or at Jenna or at Nicole in the Slido Ask Questions section. As the talks are going on, we'll review it at the end and click on the ones that you'd like to hear. And so with much ado, I'm looking forward to our very first talk by Jenna Grimshaw from Texas Tech University, looking at the genetic diversity of the threatened northern long-eared bat, Myotis septenarialis.
Hello, everyone, and thank you for joining us. Our research is on the genetic diversity and population structure of Myotis septenarialis in the United States. Now, bats all over the world face serious challenges, from wind farms, habitat loss, impacts to our vernacula, and human persecution. And in North America, bats face the additional challenge of white nose syndrome, with some colonies facing mortality rates of up to 99%. Myotis septenarialis is a North American bat that is a gleaning insectivore. It likes feeding on moths, beetles, and spiders. It can be found roosting in trees and artificial structures and will hibernate in caves during the winter. It is distributed across Canada, the northeastern United States, and parts of the central United States. It was once common, but unfortunately has recently been listed as threatened due to being adversely affected by white nose syndrome. To better understand this at-risk species, our objectives were to calculate the basic genetic statistics, such as heterozygosity and inbreeding. 2, to determine if there is one large panmictic population or multiple subpopulations. 3, to estimate the current effective population size. And 4, to investigate the demographic history of the species. So we started off with 81 individuals from 4 different states: Kentucky, Louisiana, North Carolina, and Oklahoma. We removed 9 of the samples due to low quality DNA or a high percentage of missing alleles, and retained 72 individuals for our analysis. For the DNA sequencing, we used wing punches and liver samples and isolated the DNA with Kiegan blood and tissue kit. The sequencing was done by Admira Health using a double digest RADseq protocol and used 2 restriction enzymes, EcoRI and MSBI. Our reference genome came from DNA Zoo. Now, if you haven't checked out the website for DNA Zoo, I highly suggest you do so, as they have a large variety of really unique animal genomes available for research purposes. We used a chromosome length Myotis septenarialis genome that was recently published. It had really nice long contigs, with the longest being nearly 1.8 million base pairs and an N50 of just over 200,000 base pairs in length. So the pipeline for our genetic analysis was, we first filtered out all the low quality reads using After QC, in which we removed any of the reads that had a Phred score of less than 30. Then we aligned the high quality reads to our genome using BWA and SAM tools built the loci with GSTACS, and then STACS population program calculated all our population level statistics. Those statistics that we were interested in were nucleotide diversity, observed and expected heterozygosity, most frequent allele frequency, inbreeding coefficients, and population differentiation. So for the population structure, we used a three-pronged approach. First, running a PCA and R, then using fast structure for subpopulations k equals 1 through k equals 10, and then structure for k equals 1 through k equals 6. To determine the effective population size and demographic history, we used FastSimCol2. Now we used two different mutation rates that we were found in the literature, 2.2 e to the negative 8 and 2.36 e to the negative 9. And then we used 3 history population models. So for each of the rates for a total of 6 different models. Those 3 demographic history models we were interested in is one of the species having a constant population size, or the second model was looking at a consistent growth or decline in the population size, or having a historical bottleneck. So for each of these models, we performed the analysis for both mutation rates, so we had a total of 6 models. So for the results of our DNA sequencing, we averaged a little over 2.5 million reads per sample with a per sample coverage of 6.5, genotyped nearly 1 million loci, and then after all of our filtering, ended up retaining around 144,000 loci, with a mean locus size just under 280 base pairs. Looking at the statistics for the set the tree analysis across the 4 states, they were all very, very similar to one another. Nucleotide diversity was very low, ranging anywhere from 0.05 to 0.08. The most frequent allele frequency was very, very high, around 95 to nearly 97%. Observed heterozygosity was very low, 0.04 to 0.05, and inbreeding coefficient was quite high, 0.096 to 0.115. Looking at the genetic differentiation among the 4 states, the genetics was very, very similar for the bats in all 4 states. There just wasn't very much differentiation. And the PCA went along the same lines. So here we have Kentucky is in red, Louisiana is in blue, North Carolina is in green, and Oklahoma is in orange, and they're just really all mixed up and on top of each other. There's not any obvious clustering. Fast structure came out along similar lines. So for this particular plot, the x-axis is the subpopulations k equals 1 through k equals 10, and the y-axis is the likelihoods for that each subpopulation. The subpopulation that had the largest likelihood was 1. Now, we had really hoped to have all of the analyses wrapped up, but structure and FastSimCol2, which is doing the effective population size and historical demographics, is currently still running as of this recording. So hopefully we get those results soon. So overall, we found very low genetic diversity. We had low nucleotide diversity, low heterozygosity, high inbreeding, and very little genetic differentiation or population structure. Now, the low genetic diversity and the high inbreeding can put septotrianalis potentially at higher risk for extinction. Now, having one panmictic population rather than multiple isolated subpopulations, as well as having a large geographic distribution area, might be the saving grace for this species. Specifically, Louisiana could be a vital refuge for the species. So far, no evidence of white nose syndrome or the fungus that causes it has been identified in Louisiana, although it has been positively identified in surrounding states. So the septotrianalis in Louisiana does capture a lot of the genetics of the species as a whole. I'd just like to take a moment to say a special thanks to everyone in my labs, the David Ray Lab, as well as the Richard Stevens Lab, Louisiana Wildlife and Fisheries, and USDA, and all of the organizations that made this project possible.
Hello, I'm Zixia Huang from University College Dublin. Today I'm going to talk about my ongoing project, investigating the molecular basis of extended longevity evolved in bats. I will present some preliminary results and demonstrate how high-quality genomes are crucial to better our understanding of mammalian aging. So human life expectancy has dramatically increased since the 20th century. It is predicted that the baby born today could live to be the age of 142 years. That's great news, but for humans, living longer and longer causes problems. This is because despite an increase in lifespan, the onset of the age-related diseases such as cancer and dementia has remained unchanged. So the goal of aging research is to figure out how we can extend health span so that we can live longer and healthier at the same time. Bats are the longest lived mammals given their body size, showing little signs of aging. They are even more aging resistant than humans, which makes them ideal models to understand the molecular basis of healthy aging. To compare the maximum lifespan across mammals, the longevity quotient was introduced, which is defined as the ratio of observed longevity to expected longevity of that size. So according to this, 19 species of mammals live longer than humans, 18 of which are bats. The only exception is the naked mole rat. This suggests that bats may have evolved unique protective mechanisms to combat the aging process. So to study aging in bats, we need to find a suitable long-lived population. Luckily, we have worked with our amazing collaborators, Britain Vivant, during the past 10 years and established a new study population of long-lived Myotis myotis bats in Brittany, France. These little creatures only weigh around 20 grams and can live for up to 37 years. We have developed a new approach to non-lethally sample these bats year after year as they age, so that we can identify the age-related gene expression changes that may underlie the molecular basis of healthy aging in bats. In my previous projects published last year, we deep sequenced the 150 blood transcriptomes and microRNA from 17 individuals ranging from 0 to 7 years old. Sequencing transcriptome and microRNA at the same time enabled us to explore how gene expression is regulated during aging in bats. We performed a systems-level analysis by investigating the age-related expression changes of protein-coding genes and microRNA, and further comparing their patterns to other mammals. So what did we find? We have uncovered the transcriptomic signatures of longevity that are specific to bats. The pathways associated with DNA repair, autophagy, and tumor suppression showed positive correlation of gene expression with age in long-lived bats, a correlation not seen in humans and mice. However, comparative genomic analysis has revealed that most of these age-related genes are under purifying selection. This implies that it is gene regulation rather than gene sequence that plays an essential role in mammalian aging. So we look at microRNA regulation first. MicroRNA are a class of short non-coding RNA that can specifically bind to the 3' UTR of target genes, thus suppressing gene expression. By target prediction and co-expression analysis, we found that some genes implicated in the aging process were likely to be suppressed by certain microRNA, as they exhibit significant negative correlation, as you can see here. We further successfully validate some pairs in human cell lines using luciferase reporter assay. This indicates that the longevity signatures observed in bats were regulated by microRNA. However, we also noticed that a large proportion of the age-related pairs did not show significant negative correlation, as you can see from these examples. It is apparent that other important regulators of gene expression were also engaged in this process. This result stimulated the hypothesis that bats' longevity profiles are subjected to a multi-layered post-transcriptional regulation that involves an intricate interplay among diverse RNA species. So in general, it has been shown that other non-coding RNAs, such as long non-coding RNA, circular RNA, and pseudogenes that contain microRNA binding sites can co-regulate each other by competing for shared microRNAs. We call them RNA crosstalk. RNA crosstalk has been reported to regulate a series of human diseases. For example, pseudogene PTEN-P1 shares 5 conserved microRNA binding sites with PTEN, a tumor suppressor, and plays major roles in cancer progression, while circular RNA7 contains 60 binding sites for microRNA7, thus modulating the expression of its oncogene targets. As microRNA-mediated regulation cannot fully explain gene expression changes, we speculate that RNA crosstalk is involved in the regulation of aging in bats. So how to predict RNA crosstalk? We have employed a comprehensive pipeline to predict RNA crosstalk from massive transcriptome data. In brief, real RNA pairs usually exhibit a co-expression manner, and at the same time, they shared a significant number of microRNA binding sites. Assessed by hypergeometric tests and correlation analysis, we are able to predict putative RNA crosstalk, which is subjected to further functional validation and cellular assays. Here are the 2 examples. Clock gene, a transcription factor controlling circadian rhythms and aging, shares 4 microRNA binding sites with meiosis link RNA15. Similarly, IGF-1R, whose deregulation will cause cancer in humans, shares 5 binding sites with link RNA24. Both pairs are likely to be putative RNA crosstalk, as evidenced by hypergeometric tests and correlation analysis. These pairs are the reliable candidates for downstream functional experiments to evaluate their phenotypic consequences in cellular aging, of which analysis is still ongoing. So by uncovering RNA regulation during bats aging, this project will drive a better understanding of the causal mechanisms underlying extended longevity in long-lived mammals. This may provide novel molecular targets that could potentially be modified to alleviate human aging in the future. Last, I would like to stress how high-quality genomes are crucial for this project. We need them to identify and quantify all types of RNA and to determine whether these RNAs are species-specific. We also need to obtain 3' UTR sequences of messenger RNA, and thus to predict putative RNA crosstalk. Finally, we need them to conduct interspecies comparison to determine whether the RNA regulation that underlies healthy aging is unique to bats. Without high-quality genomes, all of this will be extremely difficult, if not impossible. So I would like to thank my supervisor, Professor Matilin, and all the amazing collaborators contributing to this project. Thank you for your attention. I'm happy to take any questions.
Xenopterus brachyotis is a fruit bat widespread in Southeast Asia, ranging from Sri Lanka, Thailand, Vietnam, Indonesia, Malaysia, and the Philippines. The species is believed to reach the Philippines within the past 1 million years. Currently, it is abundant in different types of habitats such as agriculture, residential, and forested areas. Because of their abundance and widespread geographical extent, the species is not a priority in conservation. The status of Xenopterus brachyotis taxonomy is uncertain and is considered a species complex, which could be possibly composed of 7 species, such as Xenopterus brachyotis and Xenopterus luzoniensis. But data of some of these species on its status and distribution are still unknown. In the Philippines, there could be 2 possible species within this complex, which includes Xenopterus luzoniensis, which is present throughout the Philippines and Sulawesi Island of Indonesia, as shown in the figure, and the Xenopterus brachyotis, which is possibly present in the island of Palawan. However, authors all over the Philippines still use Xenopterus brachyotis and do not place the population in the country as Xenopterus luzoniensis. Even the taxa has gained support from international researchers. This is because the content, distribution extent, and proper name of the species in the Philippines is still unclear and unresolved. Thus, this study was done. Good day, everyone. I am Lefexon Gamalo, a wildlife biologist and a member of the Philippine Genome Center Mindanao, and I'm going to present our initial result of our research entitled The First Complete Draft of Mitochondrial Genome Sequence of Cynopterus brachiotis from Mindanao, Philippines. Our team in the Philippine Genome Center Mindanao aimed to generate the full mitochondrial genome sequence of the Philippine Cynopterus brachiotis, which can be used to answer the unresolved problems of the taxonomy of the species in the Philippines. We also aim to establish the evolutionary relationship of the Philippine Cynopterus brachiotis to its Asian counterparts through mitogenome-based phylogenetic analysis. To do this, cardiac muscle was obtained from a voucher specimen of Cynopterus brachiotis morphologically identified using the BAT taxonomy guide of Engel and Healy. Genomic DNA was extracted using the Quagen DNA-C blood and tissue kit according to the manufacturer's instructions. DNA enrichment was performed through a long-range PCR using two primer sets designed to amplify the BAT mitochondrial DNA. Library preparation was performed using Nextera XT DNA library prep kit, and libraries were sequenced using the Illumina MySeq platform. Here is the overall workflow of the bioinformatics analysis. We use FastQC and Trimonatic to process the raw reads data. To assemble the quality filtered reads into a whole mitogenome, Myra and MitoBeam was used. Mitose and nucleotide blast was then used to annotate the whole mitogenome of the species. After this, mitogenome sequences of other fruit bat species available at the NCBI database were aligned with the complete mitogenome of the Philippine Cynopterus brachiotis using MEGA 10. A maximum likelihood tree was inferred in MEGA 10 using the best substitution and heterogeneity model calculated from G model test 2 with 1,000 bootstrap replicates. From the analysis of the data, 653,967 clean paired end reads were used to assemble the 17,382 bases length of consensus sequence with 41.48% GC content. These statistics are comparable with other published mitogenome of bat species online. Mitose online server was able to resolve all mitochondrial genes in the genome except for one transfer RNA gene, which may be further resolved by additional capillary sequencing of the region. Sequence analysis showed that the Philippine Cynopterus brachiotis is only 90 to 91% homologous with other Cynopterus species based on its full mitogenome sequence. Phylogenetic analysis of fruit bat mitogenomes deposited in GenBank revealed that the Philippine Cynopterus brachiotis in this study have diverged from Asian Cynopterus, as both the Cynopterus brachiotis and Cynopterus lynx from other parts of Asia form separate clade, although all Cynopteran bats form a monophyletic clade. This divergence at the species level was consistent with individual gene trees using the cytochrome oxidase 1 and cytochrome B gene markers. Our results support previously reported hypothesis that the Cynopterus brachiotis in the Philippines is distinct from its Asian counterparts and should be elevated to a new species. However, our result does not conclude that the Cynopterus species in this study is a Cynopterus luzoniensis nor a Cynopterus brachiotis. Clarification of the taxonomy of Cynopterus in the Philippines is a need, and there is a possibility that the Philippine forms of Cynopterus could be possibly a Cynopterus luzoniensis, or maybe a different species. Thus, a phylogenetic tree with representative samples from Palawan, Sulawesi, including and other Cynopterus species from other parts of Asia, should be generated to clarify the status of Cynopterus species in these places. This analysis should be supported by other types of tests, such as morphological and ecomorphological comparisons with other closely related taxa within this complex. Proper naming and identification should be a priority to ensure the survival of species and genetic conservation of the taxa.
Okay, so as a brief refresher, transposable elements, or TEs, are repetitive elements that can move around the genome, of which there are two main groups, retrotransposons and DNA transposons. And they have a variety of evolutionary impacts, including exaptation by the host, regulatory disruptions, non-homologous recombination, and so forth. And so they're overall mutagenic. Now, recently, Wicker et al. Showed that transposon excisions and insertions are associated with an increased number of mutations in nearby DNA sequences within rice genomes. So as you move closer to a transposition site, the number of SNPs and indels increases well beyond the genomic average. They suggested a model of error-prone DNA repair, where after a transposon excises or inserts itself, there is low fidelity DNA repair of the site, introducing a lot of mutations. Now, the TE thrust hypothesis is the idea that there is a Goldilocks zone of T activity and accumulation, where a certain amount of T activity can allow genomic dynamism and potentially rapid lineage evolution, but not enough activity to cause genomic instability. And this leads us to Vespertiliana bats. Vesper bats, unlike closely related families, have had multiple waves of DNA transposon activity and accumulation in the recent past. And this activity corresponds with the rapid diversification of this family. Vesper bats represent over 400 species, and the genus Myotis, which has additional recent DNA transposon activity shown in orange, have over 100 species, making up about a fourth of Vesper bats. So what we want to know is, are there TE-associated increases of mutations in Myotis bats? We expect that DNA transposon insertions and retrotransposon insertions will cause increased mutation rates that are higher than the genomic average. So we used the new BAT1K Myotis genome assembly and repeat masker to find all elements in the genome. We then analyzed paired end Illumina reads of 10 other Myotis species with the mobile element locator tool, MELT, to find all polymorphic transposable element sites across all 11 species. We then analyzed the genomic variants, the mutations, in DNA sequences flanking these polymorphic sites. And there were plenty of sites to look at. And we found basically no SNP enrichment, and this was consistent across all species and all T families. There was also very little indel enrichment, and this was consistent across all TEs except signs. There was indel enrichment within the first 20 base pairs flanking sign insertions. This enrichment was only within the first 4 bases of line and HAL sites. So this was completely different from what we expected. So did we screw up? Well, a colleague, Joe Manthei, did the same analysis in woodpeckers, and they showed trends more like what was found in rice, but with a still limited range within about 200 base pairs. So we're fairly confident that our results are real. So if these results are real, are Myotis bats special, or bats in general, or does this reflect a broader difference between mammals and plants? Well, if Myotis are special, there is a possible explanation. Recent studies have shown that Myotis have upregulated DNA repair genes. So maybe Myotis, or mammals, have higher fidelity DNA repair mechanisms, reducing mutations around these insertions. So there's a lot more work to be done. And with that, I would like to thank everyone on this slide, especially my lab and BAT1K. And thank you for listening. And let me know if you have any questions.
So I just want to say thank you so much to you brilliant speakers. It was extraordinary to do this online and have it all recorded. And really, I found this session so very, very interesting. And I know that we have a whole bunch of questions that are now coming in, and I can already see some of them. And what we're going to do, everybody have a look at the questions and also try and like the ones that you want me to ask. And I'm going to ask a question to each one of our speakers. And as we get more and more, I'll keep going around until the time is up. So the first question I have up in my list, it's actually for Zaja from Andreas. So it says, are these candidate RNA networks likely to be active across many cell types in the blood, or are there ones that seem to be driving the process? So Zaja, you're up.
Okay, that's a good question. So the RNA networks were mainly tested in model species. And the examples I posed in my talk are actually bat specific. So we are trying to get the most reliable candidates in terms of bioinformatic analysis to do further functional validation and to see the phenotypic consequences in cellular aging.
Okay. So a next question that I have here, again, it's a question that we're going to ask Jenna. So Jenna, on your PCA, some individuals seem to be quite divergent, though not a signal related to their geographic origin. How would you explain this?
That is a very good question, and we're looking more into some of those to see what's going on with those specific individuals. It could just be that they're missing, that their alleles are they're missing a higher quantity of alleles, something along those lines, but that's going to warrant more investigative research.
Okay. And so this next question, we have, it's bringing together two questions, and it's for Leaf. And it's from Sebastian Puschmeyer. So it's nice and important work. I'm curious to know why you decided to use mtDNA rather than nuclear DNA to investigate this taxonomy of Synopterus.
Yeah, I think that's a good question. But I think that it's a standard for barcoding identification. And we all know that mitochondrial DNA are relatively conserved compared to nuclear DNA, and it's good for tracing ancestry. Thank you.
Okay. And so, Nikki, I have a question for you. And so, Vespertilio bats, they're really, really extraordinary. And as you can see, they have these huge active transposome elements that you talked about. But I suppose my question is, why the Vespertilionids? Why are they so special?
Well, so, I mean, in general, it's just, they seem to be really interesting because they have these multiple ways of DNA transposons. And I mean, I couldn't tell you the reason why they have so many DNA transposons, but they have so many that it just makes them really interesting to look at in terms of transposable elements and how those might be contributing to their genomic evolution.
And would you even try to hazard a guess, or come on, how would we address that question?
So currently, one of my other lab members is looking into horizontal gene transfers. I mean, in this case, horizontal transposable element transfers from arthropods, I think specifically, to see if any of these DNA transposons originated in arthropods and then just sort of diversified into these bats. But so far, I don't think there's anything conclusive.
Okay, it's a cool area of investigation. Lots of transposable elements, but still very small genomes. What are they doing? So I have another question here again. So we're going back around. So Zaja, this is a question from Nicole Foley. Are the RNA species you mentioned the only RNAs that are involved in RNA crosstalk? And is it possible to query all RNA species?
Yes, any RNAs actually have the microRNA binding site can potentially have this cRNA crosstalk. Even the protein coding genes themselves can compete each other with these binding sites. So yes.
Okay. I just seen a question here, and Jenna, you're going to get this question, but maybe everybody can answer it. It's a question here from Jorns. If the bats are panmictic, and assuming the gene flow happens among the populations, what's so special about Louisiana bats?
So one of the reasons that we're looking at Louisiana specifically is there is no evidence of white nose syndrome there so far, even though there is evidence all around in the surrounding states. So surrounding states are experiencing very high levels of mortality rates. And so we're hoping that Louisiana might end up kind of being a refuge where the bats in Louisiana, will still have a lot of the genetic diversity that we see elsewhere.
Okay. So Leif, I have a question for you, and it gets back to the idea of why only use mitochondrial DNA. So I'm interested to know, do you have plans to sequence nuclear genome, different parts of different types of nuclear markers, or how are you going to really test that the phylogeny that you actually see from your mitochondrial DNA is really the true evolutionary tree and rather than just a species tree? What's the plan?
So actually, this is a bigger project. So we have done some works also in other species of bats. But so far, we only did some barcoding for this
Oh, we've lost your sound.
Specific specific gene markers, and the result is still the same. And that's a good thing to look at to also barcode the nuclear DNA.
Yeah, to try and nuclear DNA. And what do you expect that do you expect your mitochondrial tree will be upheld by looking at other genes? Leif.
I'm not sure about that.
Oh well, we can try.
Let's see.
Yes, Synopsis brachiotis. They're beautiful, beautiful bats, and really we should think about sequencing one of these guys for bat 1K is what I'm thinking, to be very honest. So Nikki, I have a question for you. Yeah. Nikki, I have a question for you, and really it's kind of for all of our speakers right now. So the idea is that what is the benefit by having these chromosome level, really contiguously, highly accurate, exquisite genomes? I mean, Nikki, you're going to answer it first. What is the benefit of that to the question that you are asking?
Well, for me, it's definitely because it needs to be really high level, because otherwise since T's are so repetitive, they won't be actually in the genome assemblies. And so we won't have an accurate picture of what's going on and where they are in the genome, especially in terms of where they are on the chromosomes. So we need to know where they are in relation to genes in particular.
Yeah, so that's why you need the long reads. And again, of course, guys, I'm going to be pushing this. Jenny, your question is even a bit more difficult though, because there's always been this, and constantly with VGP and G10K and Tree Black, there's always discussions is, should you invest money in this highly accurate reference sequence, or should you kind of do low coverage genomes for many individuals if you're looking for some type of conservation genomics question? So how did you find it more beneficial for your question to have this a long chromosome length genome that's exquisite or not?
Yes, I did. I think with the long chromosome length genome available to align all of my reads to, then I was able to be more confident in that I wasn't losing information. I wasn't losing loci. I was able to, just the data is better. And so I think it's absolutely important to spend the time and the energy and efforts into creating these really high quality genomes that can be used for a wide variety of research projects, whether it be RNA projects or TE projects, or like mine, that's more population genomic, research. It's important to the research as a whole.
Yes. And Zaja, if you want to chip in on this, how was your research helped by having these good
Oh,
genomes at hand.
It definitely, I think, is benefit everyone, and not just looking at the protein coding genes, but also the non-coding genes. So basically, a lot of my projects are related to a non-coding gene evolution. So so the high quality genomes, is especially chromosome level genomes, can help you to recover all these non-coding genes, for example, long non-coding RNA genes, new mite, and microRNA genes. So actually, I did some straight comparison between the chromosome level genomes and the Illumina genomes, and we recover much more. So it can also help you to answer the evolutionary questions if we have the high quality genome alignment. So it's definitely helpful.
Yeah, find the regulatory regions. I agree completely. And Leaf, for you, do you see the benefit of having a really exquisite genome for, say, Synopterus brachiotis?
Yeah, I think this is very important thing to look at, because this species is actually a problematic in terms of its taxonomy, and sequencing all those genes would be helpful to solve this problem. And we all know that we cannot conserve a species that we don't know. That's why it's very important.
Okay, so guys, we're going to wrap up now, and I want to plug the next session, which is VGP. We all work together as part of group. Bats are vertebrates. The VGP group is going to talk about sequencing the genome of every single living vertebrate. And I just want to say again, congratulations to our amazing speakers. I'm just going to read out some of the really nice responses from Jose. Amazing talks, amazing work. So people are very excited by your presentations. Well done. And everybody, please join Bat1K, and together we can uncover the unique and wonderful adaptations that bats have. So thank you all.